So the question is about extracellular pathogens and intracellular pathogens. So I list a whole bunch of bacteria, and this is what I want you to know from that about those bacteria. First, remember that in pathogenesis, there are these 3 basic steps after the entry into the host in order to cause disease. So attachment is required before the bacteria can colonize. In order to colonize, they need to get nutrients. So they need to be able to get the nutrients they need to grow. In some cases, that means that they need to produce a toxin to get the nutrients they need. For example, a toxin that would release iron. That's mostly bound up in our tissues. And the third thing that all pathogens need to do is to evade our immune response, right? So when you look at this list of microbes, for the bacterial pathogens, I want you to be thinking about those key 3 aspects of pathogenesis as far as attach, acquire nutrients so that they can colonize, and evade the immune response. And only through those 3 things can they actually cause disease. And so disease, as it's defined, is any damage that's caused to the host, whether it's directly as a result of a product of the pathogen or whether it's a result of our own immune response, say inflammation. And an inflammatory response can often do damage both to the pathogen and to our own tissue. So let's look at the list. You can see here at the bottom of the page here, I have the bacteria listed. And then on the second page, I have the other microbes that I have on the list. For all of the microbes, you should know what type of microbe they are. Are they bacteria, virus, fungus, protozoa? So you should be able to classify them as such. And then for the bacterial members of that list, so the bacteria that I've listed, you should be familiar with any features that are important for their pathogenesis. So I've listed here Clostridium is an endospore former. Okay, and that's important because it can survive antibiotic treatment. E. coli, we learned about in the presentations that were made that the pathogenic form differs from the normal microbiota because it produces exotoxins. But also remember that any of these on the list that are gram-negative also have endotoxin just inherently as part of their cell wall, the cell envelope in the outer membrane. Vibrio cholera also produces an exotoxin that is responsible for the symptoms of the disease of cholera. Then Staphylococcus aureus in its pathogenic form often, but not always, forms toxins. One of which is the toxic shock syndrome toxin, but it also produces, like we saw in the lab, like it lyses the red blood cells on the blood agar plate, so it produces hemolysins. Shigella is also a toxin-producing strain of or species of bacteria. Producing the Shiga toxin. And there's a Shiga-like toxin that's also part of the pathogenesis of some strains of E. coli. And then Salmonella. So when I'm talking about intracellular versus extracellular, which was your original question. So most bacterial pathogens are going to be attaching to tissue and colonizing outside of our own human cells. But remember that we learned that some of the pathogens, the way that they evade the immune response is to have kind of trick our cells into taking up the bacteria so that it's inside our host, our own cells. And when it's inside our own cells, it's not visible. It's kind of hidden from our immune response. Our B cells can't detect it. Our T cells can't detect it. Our antibodies are not having access to that site, so they have no effect on the pathogen. So remember from our homework in chapter 18, there was an open response question about Shigella, Salmonella, Listeria, and Coxella as examples of these types of pathogens that hide out to evade the immune response. They hide out and they're intracellular pathogens. So they're inside our host cells, and that's when they colonize or are able to grow and evade the immune response or pass to another cell type where they can spread and disseminate in our body. So that's what I mean by extracellular and intracellular. The vast majority of bacteria or any eukaryotic pathogen would be an extracellular pathogen. Just like our normal microbiota, they're there, but they're not inside our cells. They're just kind of lining the tissue of the lumen of our gut or on the surface of our skin or our mucous membranes. They're not inside our cells. Okay? Does that clarify?
No, that's perfect. I was just wondering what we should know basically about all the bacteria and viruses in that question. So that was helpful. Thank you.
Okay. Any other questions? Emily, do you have a question?
No, that was my main concern, that last one, really.
So I think the big ones I already went over in class with the innate immune response. So these open response questions for the essay. Be sure that you are familiar with the details of phagocytosis and what type of cells carry out phagocytosis, those innate immune cells that are part of our first line of defense. And as I mentioned already, the intracellular pathogens, how you are able to avoid phagocytosis by hiding out in host cells, as well as by putting a stop to a key step of the lysosome fusing with the phagosome or escaping from the phagosome before that happens. Then the influenza and polio, you should be familiar with the vaccines and how they each have both an attenuated and an inactivated form of the vaccine. But with influenza, you should also go back and review the genetic features that account for these either slow changes of drift over time or large changes, and we had presentations on these topics. So antigenic drift versus a big change that leads to a pandemic strain, that would be a shift. And then what's the difference between an exotoxin and an endotoxin? When in the steps of pathogenesis would a toxin be produced? These bacteria are very conservative with their expending energy, and so they aren't always producing toxins. It's only once they have attached and colonized when some bacterial pathogens will begin to produce toxins as part of their pathogenesis. And some have to invade first. They invade the tissue first, then they colonize, then they produce toxin. And then as far as vaccines go, this is an artificial way of ideally inducing like lifelong protection from disease. So how does an attenuated vaccine work to do that versus an inactivated vaccine, and those key differences that we also went over. But I'm happy to go over any of these again if anyone wants clarification.
Can we go over that briefly? The attenuated versus inactivated vaccines, how they work?
Absolutely. So Annalise asked me to go over the difference between attenuated and inactivated vaccines. That material is covered in Chapter 17. So remember that all vaccines are an artificial way of getting immunity. What does immunity mean? You should be familiar with that definition. That just means that you are not susceptible to the disease caused by the pathogen. So you can either get sick and get the symptoms, and then you have memory cells, memory B cells or memory T cells, depending on the pathogen, or both, that help to provide lifelong immunity. Or you can get a vaccine, and that's the idea with vaccinations, is that you get the immunity without getting the symptoms. So let's look at the difference between the forms of vaccines. There are actually a lot of different forms of the inactivated vaccine. But the important thing to remember is that there are some key differences as far as safety and efficacy and how you administer the vaccine and how expensive it is and how you store it, etc. So I'll just go over this bullet summary from my slide set from Chapter 17. So attenuated vaccines are those live in that they can still reproduce, but they're weakened. So they're not going to cause disease. The idea is that often by passaging or by growing the pathogen in another species, often primates are used for this, it changes genetically to be more virulent towards that species and less virulent towards humans. But then when we use it for a vaccine, it still induces, our immune system still mounts an immune response, but we don't get the symptoms because it's been weakened. The key difference here is that attenuated vaccines reproduce. And because they reproduce, they're capable of changing again to be more virulent. So that's one risk, very low, but one risk. And then the second risk is that if you don't have a strong immune system, if you're somehow immunocompromised because you're on medication or because you have another illness like AIDS, then or you're very young or very old, and when we're very old, our immune system is waning. When we're very young, it hasn't quite developed yet. So that is, then it's risky for that population to get an attenuated vaccine because they can actually get symptoms and get sick. But the great thing and why we use attenuated vaccines is because they induce a really strong immunity with one dose. So humoral immunity is what we want to induce. We want to get memory B cells that produce antibodies in the form of IgG, or in the case of the mucosal surfaces of our gut, for example, we want to produce antibodies of the IgA type. And these memory B cells that produce these forms of antibodies will be very specific for the pathogen. So in subsequent exposure to a pathogen, then we won't get sick because our memory B cells will be right there ready to respond as fast as our innate cells are responding. The problem is the first time that you're exposed to a pathogen, you don't have that very specific antibody that recognizes that part of the pathogen that's not found on other pathogens. So it takes time for our adaptive immune response to change, and with the B cells going through genetic changes that lead to the selection of a very specific B cell that will then mature into a plasma cell and produce lots and lots of very specific antibody. It takes like maybe 5 to 7 days. And in those 5 to 7 days, that's when we start getting symptoms. And once we have the symptoms, then it takes another set of days, depending on the disease, for the symptoms to be cleared and go away as a result of the pathogen being cleared. So as I mentioned, this is attenuated. It's able to reproduce. It's very effective with one dose. It doesn't need any extra help from an adjuvant. And because, for example, if we have a pathogen that's a fecal oral route is the route of transmission, a live attenuated form of that pathogen can actually spread outside of the individual that's vaccinated to populations that haven't been vaccinated, and so it can be beneficial in helping with herd immunity. That is not the case for inactivated vaccines, which can be an entire pathogen intact, but because it's kind of frozen in time, so it's chemically treated to inactivate it. It can no longer reproduce. None of its metabolism is working. But it's intact, kind of frozen in time, so all of those antigens that would elicit an immune response are still visible to our immune system. And so that when we do introduce it into a person, it can give IgG immunity, so memory cells that produce IgG very specific to the antigens on that particular pathogen. Unfortunately, it's not good at eliciting mucosal immunity, so no IgA is produced. No memory cells that have produce IgA, and because it's inactivated, the immunity is poor or requires additional exposures to really get a strong memory response. So the memory may wane after a while, and then you get a booster, and then it's up again and wanes and up again. And then we've figured out for various pathogens how many boosters we need until we get that lifelong immunity. But multiple doses will definitely be required for an inactivated form of a pathogen. The good news is we can use it in those people who are immunocompromised, the very young, the very old. The very young is a little tricky because you have to wait for their immune system to develop long enough. So even if you give it to them when they're very young, you'll have to give a booster after age 2 so that their immune system reaches that kind of lifelong sustainable memory and sustainable protection. It does need some help. Often if you use just part of the pathogen, you'll need to add adjuvant to get it to really strongly activate an immune response. If it doesn't activate the immune response strongly, then you won't get any kind of protection from that vaccine. And ideally, we want to have 99% of the vaccine producing immunity, right? So out of 99 people, out of 100 people that are vaccinated, we want 99 of them to get, or better, to get the lifelong immunity and that protection from that disease. And then there are a couple other minor things where it would be more expensive to have an inactivated vaccine administered because you can only administer by injection. The most effective way to inject vaccines is into the muscle. And that way it really elicits a really good immune response. But it's stable at room temperature, so it's easier to store than an attenuated vaccine. So any questions about those differences? Does that answer your question, Annalise?
Yeah, thank you.
Okay. So that, as you read about in your book, there's some really interesting forms of vaccines that have been developed more recently, like just a piece of DNA from the pathogen can sometimes be enough to elicit immunity. But I'm not going to expect you to know all of those details on the exam, but I do expect you to know the details of the differences between these forms. All right, let's see. Let's go back to study guide. So a lot of the study guide is asking you to learn about pathogenesis and the immune response and kind of this balance between the pathogen and its focus on reproducing and surviving in the host long enough to reproduce, and then our own immune response to the pathogen, which is, focused on clearing that pathogen from our body. So you should know not just about the innate immune response, because pathogens are quite good at evading the innate immune response such that we get symptoms before the adaptive immune response is developed. But you should also know, so in the innate immune response, you should know the barriers, the key cells, phagocytosis, and then the key chemical factors like the complement that's constantly circulating our blood, how that's activated. Antibodies will circulate our blood, but that's only after the adaptive immune response matures. What else? All the steps of phagocytosis, including the key step of the lysosome fusing with the phagosome. And once that happens, the contents of the lysosome are in contact with the pathogen that's been engulfed. So exposure of the pathogen to those factors, which include enzymes that are destructive or lytic, and very low oxygen levels, also oxygen radicals, which can be toxic to some bacteria. So there's an oxygen-dependent mechanism of killing in the phagolysosome, and there's an oxygen-independent mechanisms of killing. Low pH also can kill the pathogen that's been engulfed into the phagolysosome. And let's see, as far as adaptive immunity goes, remember the B cells are the ones that produce the antibodies. You should know the 6 mechanisms of action of antibodies and how they function once they're secreted from a plasma cell and are circulating in our tissue or in our bloodstream to the site of infection, what they do to a pathogen to help clear it or destroy it. Let's see. Also, you should know the different types of T cells. So the T helper cells are helping not only the T cells, the cytotoxic ones, but also the B cells in activating them to generate a specific immune response that's specific to that particular pathogen. And then the cytotoxic T cells are the ones that are attaching directly, contacting. The entire CTL is directly contacting an infected cell. This is where intracellular pathogens are how they're cleared by cytotoxic T cells. And the T cell can detect a signal from our host cell. Usually our host cell will not elicit any kind of response. But in the case of say a virally infected cell, out on the surface, the infected cell will present little bits and pieces of the virus so that it will be a signal and it presents them inside the MHC molecule. So all cells have, almost all cells have MHC class 1 that is able to present these viral bits and pieces to the cytotoxic T cell. And once the cytotoxic T cell recognizes that, only the one that's specific for that antigen would be responding. But once it responds and binds to that sick cell, host cell, it'll be able to induce apoptosis. And that's by secreting enzymes that are degradative, so granzymes and other enzymes that will kill the host cell that's infected. So that's intracellular pathogen. Extracellular pathogens, like most bacteria, are taken care of mostly by our B cells that produce antibodies, which then interfere with their function. Things like, let's see. So remember here you can see the structure of the antibody. This part in yellow here is the variable region that is able to bind and recognize a specific part of a pathogen. And that is the part that changes as the adaptive immune response proceeds. And then once you've generated specific antibodies, then they can neutralize or immobilize antigens or clump them together through agglutination or signal for the innate immune response, the phagocytic cells, to engulf and destroy them, or activate the innate chemical signals of the complement, so that they in turn mark the either directly kill the pathogen in case of the membrane attack complex forming, or they can also help to signal for phagocytic cells to destroy the pathogen. And then for really large pathogens like multicellular pathogens or eukaryotic pathogens, natural killer cells come in and play a role in, similar to a cytotoxic T cell, where it's secreting enzymes that are destroying not our own host cell this time, but the pathogen. Any other questions about that material?
Can we go over those mechanisms of action for the antibodies? Then, like, are you talking about like the IgA versus IgG, or you think that's something different?
So whether it's IgA or IgG, it's really going to have a very similar function. IgM, because it's a pentamer, it tends to be much better, say, at agglutinating pathogens, because you can see in this figure to the right, let me just move it down a little bit. We're only, the red is the antibody, and that's just a monomer, similar to how you would see IgG acting. But any of these other classes of antibody can have similar functions to a monomer. The only thing that a dimer or a pentamer would not be able to do is opsonization, because they're the kind of stick of the Y. So you have the Y, and the two arms are out there binding the antigen, and this heavy chain, which is not variable, it's like a fixed region, is in a dimer, it's not available, or in a multimer like a pentamer, it's not available to be recognized by the receptors on a phagocytic cell, or recognized by complement. So only IgG would be able to carry out opsonization or complement activation, or NK cell activation. But the other forms, IgG, IgA, can carry out neutralization or immobilization or agglutination. Does that answer your question?
I think so. I was talking about before, when you were talking about the 6 mechanisms of action for antibodies. Were you talking about something different, or were you talking about like neutralization, immobilization,
Yeah,
agglutination?
those are the 6 protective roles. Those are the 6 mechanisms of action of antibody are the 6 protective roles here: neutralization, immobilization, agglutination, opsonization, complement activation, and antibody-dependent cell activation for in the case of the NK cells. Any other questions about any of the material on the on the study guide. Christabel, I see that you've joined. Let's see who else. Does anyone else have any questions? I'm not hearing anyone because I think you're all muted. But if you unmute yourself, I'll be able to hear you.
Can we talk about like the specific changes in influenza and why it's necessary to make a new vaccine every year specifically?
Sure.
Yeah.
Let's see. So that would be in chapter 12. So in chapter 12, we learned about viruses in general, but there's also a part where I was talking about influenza specifically. And you had a open response question about antigenic shift and antigenic drift. So let's just look at the virus itself here on the right. So the structure, as we learned about from, I think a couple people did a presentation on influenza. But just to refresh your memory, there's the way that the flu vaccine, that the flu virus itself is organized as far as its genome, has a lot to do with why we have pandemic forms. But your question was about the seasonal flu, and so the key here is that influenza has an RNA genome. Any virus with an RNA genome is inherently more likely to acquire mutations and inherit those mutations in the sense that the virus progeny will be different from the parent virus that infected that cell. So remember that any virus, in order to reproduce, it has to get into a host cell because it doesn't have the machinery, it doesn't have energy, first of all. It has no way, it doesn't have a metabolism, so it can't produce any ATP. And ATP, even if it has its own RNA polymerase, such as the case with the influenza virus, it won't be able to function without ATP being present. So it has to get in the host cell. Outside the host cell, it's dead, it's inert, harmless. But once it gets in the host cell and starts to reproduce, that's when it's really going to result in this damage and to our tissues and the signs and symptoms of that disease. Okay, so what's special about RNA? Why is it less stable than DNA? It has to do with the polymerase. So our own cells have RNA polymerase, which some viruses do use, and in the case of influenza, it has its own RNA polymerase. The RNA polymerase, though, is very different from the DNA polymerase. Remember, the DNA polymerase in our own cells is there to make sure that it's making a copy and an accurate copy of our genome. When our cells divide, we don't want to create cancer cells by having a lot of mistakes in the genome, right? So our DNA polymerase has this proofreading function where it goes back and checks its work before the cell cycle can progress before that completion of making another cell. It's not the case for viruses. Viruses don't have those checks and balances, and our own. So if it's a viral RNA polymerase, it won't have that proofreading. And if it's a host RNA polymerase, it also doesn't have that proofreading capability. So it makes a lot of mistakes along the way. And those mistakes, as long as the protein components are present in the host cell to make the kind of build a new virus structure around the viral genome, it's going to be made into a new virus. So these mistakes are random mistakes. Sometimes they result in a virus that no longer can replicate, and so it even if the even if it gets into a new host cell, if it can't replicate, then it's not going to be causing damage and disease in our body. But some of those other changes change it just a little bit, but it's still able to get into our other cells and replicate. And so then when it leaves our body, it's kind of in a different form than when it came in our body, right? So when it's being transmitted and exiting our body and going to find a new host, it's in a different form, even if it's slightly different, because these mutations won't make a large difference like a pandemic flu, but they would make a small difference. And you can think of each time it goes into a person, it changes a little bit more, so that across the entire season of the flu season, what you had at the beginning of the flu season and at the end is actually quite a different virus. So we do our best to make an educated guess about what the key strains of influenza are, but we have to make that guess about this time of year, like May, June, in order to have enough time to have the manufacturing process of the flu vaccine ready by, late August or September for the next flu season. And during those months, it can also change even more because the flu goes to the southern hemisphere where there's winter when we have our summer. So those small changes are what accounts for now the next flu season. Even if you got sick with the flu this year, you won't have full immunity to that next version of the flu virus. Does that make sense? So it would just be individual bases within the genome that were changed, not a reassortment like we see with the mixing of two viruses in a single cell, like with the swine flu. That's different. The seasonal changes are just mutations, point mutations that are just made because the RNA polymerase makes mistakes along the way.
And is the mixing what would occur for a pandemic flu?
Yeah, that's where you have to have two viruses in one host cell. Otherwise, it won't happen. Okay, it has to be in the same host cell, and that host cell is now its machinery and energy is being hijacked by both those flu viruses. And then when it goes to package these multiple segments of the RNA genome, it may accidentally take, say, a pig flu virus segment and mix it with a human flu. And then you get a very different human influenza virus. It can still infect humans because it's got the correct hemagglutinin molecule that is like the key to get into the host cell, our host cells in our upper respiratory tract. If it loses that segment, it won't be able to infect humans. Or if the swine flu or the pig flu gains the segment that encodes the protein for the human recognizing form of hemagglutinin, then it can jump species from the pig to the human. But this is a very rare event. You really have to be working with these animals in close contact with them to be exposed to such a rare virus. Either birds or pigs can be a source of this pandemic flu.
Is there a specific diagram from like the book or somewhere that we should use for labeling the steps of like a phagosome?
The steps of
Oh, sorry.
phagocytosis should be in chapter 15. This one is pretty good.
Yeah, that's the one on page 461, I think. Yeah,
Page 461.
yeah.
Yep.
Just double checking.
Yeah, it has all the necessary steps of like, recognition and adherence. The phagocytosis, which is the ingestion step, which just forms a phagosome, but then you can see how the lysosome that already exists in the cell and contains those lytic enzymes. So the lysosomes have other functions in our own cells. All of our cells have lysosomes because they're kind of like the recycling center of the cell where they kind of break down stuff and recycle it for use in biosynthesis. But it's used very effectively by phagocytic cells, which are the neutrophils, the macrophages. Monocytes mature into macrophages when they get in the tissue. Also, dendritic cells can carry it out, but the two main ones, like the major immune cell that's really in the highest numbers, are the neutrophils that carry out phagocytosis. So once that fusion step occurs where you get the phagolysosomes formation, that's when the contents of the lysosome, as far as the reactive oxygen species, the lower pH, the lack of oxygen, the lytic enzymes, those will all help to break down the physical structure of the pathogen, whether it's bacteria or virus. And once it's broken down, the virus can't infect any cells anymore, and the bacteria can't, it's dead, so it can't attach or colonize or anything. So it's harmless, and then the contents are released by exocytosis, which is kind of the opposite of how it gets in. It gets in by endo, meaning inside, cytosis, inside the cell, and out by exocytosis. So yeah, if you know those steps, then you're all set for that. I have them written out here to the right.
Thank you.
So we have about 10 more minutes. Anyone else have any questions? Okay.
You want us to specifically talk about how like Shigella and Salmonella avoid phagocytosis? Like those
Right,
specific.
that's the pathogen part of their pathogenesis. So remember in chapter 18, I asked you to, as part of the homework, so the question was about how Shigella and Salmonella are avoiding phagocytosis. And so remember we had an open response question as part of the homework, and you can go back and review my comments on that. But basically, There are several pathogens, not just Salmonella and Shigella, but also Coxiella and Listeria that are able to hide out from the immune system. So remember, in general, there are three key functions that a pathogen wants to achieve in order to cause infection and disease. One, it has to attach to the host cell or host tissue. Two, it has to obtain the nutrients it needs in order to colonize or reproduce. And then three, it needs to evade the immune response long enough so that it can reproduce. And so some pathogens are able to do that. Here's kind of a nice summary, actually, of different factors that make their virulence factors that make a pathogen different from a harmless type of bacteria that won't be able to cause disease. But let's look specifically at your question as far as these subset of bacteria that are actually intracellular pathogens. So I mentioned before that most bacteria, they're attaching and colonizing outside of a host cell, but some are able to get inside a host cell and survive that way, and that's the way that they evade the immune response. I don't think I have that picture here, do I? Nope. So I don't have a picture. You have a picture in your textbook about this. The one I have here is the example of Shigella. And Shigella has it's a great example of a pathogen because it has so many of these strategies to survive and reproduce in us as a host. And it only takes a very low infectious dose to do that. But basically, what the Shigella do is they trick our non-phagocytic cells into taking them up. So for example, once they get into the lumen of the gut, which is here on the top of this image, they will attach to an M cell, for example, and use a type 3 secretion system to induce a change in the cytoskeleton that just kind of engulfs the bacteria and brings it into the cell. Once it's in the cell, it's hidden from our immune response. And then in Shigella, it's in the M cells, it's actually kind of transported from the lumen to the submucosa. The advantage of a pathogen reaching the submucosa is that out here in the lumen, what else is in the lumen with the pathogen? Well, there are a ton of other bacteria that are just part of our normal microbiota. So there's a lot of competition for those nutrients that the pathogen needs to survive and colonize. But down here in the submucosa, no normal microbiota are there. At least they're not supposed to be there. And so it has very little competition. What are there are our macrophage. And so it will be engulfed, but it's able to escape the lysosome by producing cytotoxins that kill the macrophage. And when it's released, it goes into the epithelial cells nearby, and it just goes from cell to cell by this actin. So it hijacks the actin that's already in the epithelial cell lining the gut, and it transfers from cell to cell in that manner. And within those cells, it's able to use the nutrients that are present in those cells to grow and colonize and survive. So that's its pathogenesis, its way of hiding out from the immune response inside our cells, thanks to its ability to basically survive phagocytosis or avoid destruction. So the other pathogens that we had in the question for the homework have different mechanisms. So there are basically three mechanisms. The Coxiella can, I don't know why this is, okay, here we go. Let's just look at that homework because it has a nice figure. That's pretty, illustrates the point pretty well. Okay, so here's the image. I'll make it a little bit bigger. If I can zoom. Ah, there we go. Yeah, so the Coxiella is able to survive, and it actually has this unusual replication cycle in these compartments called inclusion bodies. But for the Salmonella and the Shigella, or the Listeria, I already mentioned the Shigella and the Listeria. For the Salmonella, it gets engulfed by a macrophage, which then travels via the lymph to a draining lymph node. And that's how the Salmonella will get disseminated in the body by the bloodstream. So it kind of tricks our own immune system because the macrophage here in the tissue below the gut lumen, because it can avoid destruction by that macrophage, it actually uses that as a vehicle to spread in our body. Does that answer your question?
Yeah.
So yeah, here the motility of the Salmonella is really key. It doesn't have to use the actin of the host cell to move because it has its own flagella that enable it to move. So it's another virulence factor that Salmonella have. All right, let's go back one more time to the study guide. So any last questions? Okay, if that's it, then I thank you